Excellent. Well, thanks very much for inviting me to talk in this series. It's a bit of an experimental series, as I understand, in trying to look forward to the future and not just back to what we've done in the past. So what I've tried to do is blend in some of the forward thinking throughout the talk and then have a few slides at the end where I kind of summarize things into perspectives. So the title of my talk is Seeing Early Cancer in a New Light, and that's because we use optical technologies in the study of early cancer. So I thought I'd start by just introducing to you the research in my team and then go through into my presentation. So in my team, we're interested in using multispectral imaging approaches combined with computational analysis to study tumour evolution. What do we mean by multispectral? Well, imaging, we typically think about having 2D or 3D spatial information. In multispectral, what we do is we add other dimensions of information to that. And typically it's called multispectral because we're looking at wavelength information, but we also look at other properties of light such as phase and polarization. And we take all of our data longitudinally over time to monitor tumours as they're evolving. So there is an added dimension of time there. So it's essentially taking those standard 2D or 3D dimensions of information that you would have in an image and adding the fourth, fifth, sixth or other dimensions to that. We do this using photoacoustics, which allows us to go deeper into tissue as a hybrid technique between optics and ultrasound, or using endoscopy, where we go directly to the site of interest and excite our contrast. We apply latest innovations in optics and photonics, as well as approaches from biophysical modeling and machine learning, which allow us to connect the imaging signals that we are measuring with the underlying biology. And we apply these in preclinical studies, in mouse cancer models, and also in patients. So the research I'll talk about today is really focusing on our work in endoscopy and then looking at some of the first in human clinical trial data in the context of some of the forward thinking that I want to be able to do with you. So I'll start by explaining why I think optical imaging is very powerful in the detection of early cancer, and then explain one particular approach that we've been developing around capturing the additional dimension of wavelength, so spectral endoscopy. And then finally, I'll come to some of the results from our latest first-in-human trials and perspectives on future applications. So starting then from optical imaging, I thought maybe start one step backwards and why is early cancer detection important? Well, these are some statistics from Cancer Research UK, which are a few years old now, but essentially show that early diagnosis of cancer can improve long-term survival. So across 8 different cancer types that are combined, if we look at diagnosis at stage 1 or 2, so when the disease is still confined, we have about an 81% chance of having a 10-year survival. Whereas if the disease is diagnosed late, so when the disease has already started to invade or spread to other sites, we only have about a 26%. So survival is more than 3 times higher when cancer is diagnosed early. And this is in the UK, a large scientific mission proposed by the government and also a real driver for Cancer Research UK to try and shift the stage at which people are diagnosed and really be looking at monitoring well people for signs of early cancer as opposed to diagnosing people when they're already symptomatic. So if we think about how we diagnose cancer early, there are of course a combination of tests that can be used. And typically when we think about screening programs, particularly in the UK, we'll be thinking about doing X-ray mammograms for breast cancer, or doing sigmoidoscopy for colon cancer. And there are other kind of surveillance programs where we take images from high-risk patients, for example, looking at X-ray CT for lung cancer and endoscopic surveillance in the upper GI tract, in the esophagus, for patients who have precursor conditions that give them higher risk of developing esophageal cancer. And in parallel with these tests, there are lots of tests that are being developed as well for application in the blood, and I'll talk more about those when we get towards the end of the talk. So one of the challenges that all of these early cancer diagnostics throw up is that we need to identify the most aggressive disease and treat it appropriately. So some cancers that we detect, particularly, for example, in the breast, can be relatively indolent and slow growing, whereas others can be extremely aggressive and fast growing, and we run the risk of going on to kill the patient if we don't treat it quickly. Whereas in these more indolent disease, sometimes it can be better to watch and wait, observe the disease before going in with a radical treatment, a surgery or a cytotoxic chemotherapy, which can have lots of adverse effects. So where does optical imaging play into all of this? Well, optical imaging scopes are used in a wide range of diagnostic procedures for early cancer detection. I already mentioned endoscopy in the upper GI tract and the lower GI tract. We can also examine parts of the lung And we can use it to examine various areas also in cystoscopy and in colposcopy as well. And in addition to the application of these optical scopes, there are also surgical interventions such as robotic surgery and surgical microscopy that use optical imaging to visualize tissue. What are we actually doing when we're using these optical imaging scopes? Well, we're essentially replicating what our eyes can see when we look inside the body. So we take a typical color scene and we observe that with our scope. Our scope will have some means of measuring color, which will either be a pixelated detector with an array of a color filter array on top of it that replicates the blue, green, and red cones of our eye, or it might be a monochrome camera with a light source that alternates between red, green, and blue as well. Either way, we're going to compose a series of either grayscale images sequentially with the light source modulation, or we're going to pixelate a grayscale image according to the color filter in the case of having an image sensor like this. We'll then false color that information according to the pixel that it came from and recompose our color image, which in this case will be slightly lower in terms of spatial resolution, but will have appropriate color fidelity. The challenge that poses is it actually provides relatively little contrast for early cancer. So I'm just going to give you some examples from a case study that I'll use throughout the presentation, which is in the early diagnosis of esophageal cancer. This is a disease in the food pipe, which can often emerge from sites where there are precursor lesions. And one example of a condition that can give rise to these is Barrett's esophagus. And this is an inflammatory condition for patients who suffer from chronic reflux, where the acid from the stomach is repeatedly passed up into the lower esophagus. And it causes the cells to mutate and become much more like the goblet cells here. So they become much more columnar, which is how the stomach lining looks, as opposed to these flat squamous cells at the bottom here. If you just have Barrett's, you have about 0.5% per year risk of going on to get esophageal adenocarcinoma. So that's an elevated risk compared to the general population. So these patients are invited to come in for endoscopy on a 3 to 5 year basis. And in addition to endoscopy, they also get repeated biopsies throughout the tract of the Barrett's to check for potential areas of dysplasia. That's the next grade of disease that comes along after Barrett's. So some areas of Barrett's can evolve into dysplastic lesions, which are a precursor to the development of cancer. And once your Barrett's region has one of these dysplastic lesions, it increases your chance of going on to get esophageal cancer by up to 30% per year. So this is a really huge increase. And what we really want to do is identify these lesions, because what we can do with our endoscope when we go down and visualize them is that we can then actually curatively resect them. So the endoscopist can put down some biopsy forceps in the accessory channel of their endoscope, and they can actually cut these regions of tissue out to do a very local, minimally invasive, effectively surgery, but very, very small. And by cutting them out, those lesions can then they're not present to go on to progress. So they prevent that patient from going on to get esophageal cancer. The challenge is that visualization of these precursor lesions is extremely difficult. And I'm going to give you some examples here why standard of care endoscopy really fails to provide adequate contrast. Essentially, when you look down at the tissue here, you're seeing a pink tissue lining. And when you develop one of these precursor lesions, you have just a slightly different shade of pink from the normal tissue. So if you look at these examples here, I'm going to just highlight where the lesions are. There's one over on the right in the first one over on the left in the second one, and one over on the right in the third one. So as I'm sure you can appreciate by looking at these, it's actually very difficult to discern these relative to the normal surrounding structures. And it takes many years of experience to be trained to spot them. And even then, with the really experienced clinicians, you can still have extremely high miss rates during the endoscopic procedure, which is why it's coupled with a biopsy procedure as well. So patients will have biopsies in 4 quadrants throughout their tract of Barrett's, in addition to having their inspection by the endoscope. Now, this pink colour from the tissue comes from the absorption of light by hemoglobin. And hemoglobin within red blood cells is a strong absorber of light, and the conformation of the hemoglobin molecule changes with binding of oxygen, which means that we actually get a differential spectrum whether light is absorbed by oxy or deoxyhemoglobin. And this is the spectrum across the visible and the near infrared range. So you can see actually in the visible region, the blood is very strongly absorbing. Why is that of interest in the context of cancer? Well, solid tumours require a vascular network to obtain oxygen, nutrients, and then remove metabolic waste. And their vascular network is actually very distinct from that of normal tissue. This is an example of from a quite old review now, but I really like the way that it highlights the differences between normal tissue architecture, where you have an arterial supply and a venous drainage in a highly organized capillary network, as opposed to a tumour where you'll have a very inefficient orientation of vessels, where the tumour will have stimulated the surrounding vasculature to grow towards it and will have vessels growing in from all sides. The tissue will have a high oxygen consumption rate and a steep longitudinal oxygen gradient as you move along those vessels. And often you can have a very poor vascular density as well. So the delivery of oxygen to that tumour will be both limited by perfusion of blood into the tumour and then by diffusion of oxygen out from the haemoglobin in the red blood cells to the surrounding tissue. And we know that in the tumours where we have poor delivery of blood, we'll have oxygen starvation, which generates a hypoxic stress, so a stress that arises due to the absence of oxygen. And this is strongly related with poor outcomes across a range of different cancers, and I'm just showing you some data here for breast cancer. So if we go back to our absorption spectra here, if we are able to better understand the vascular network, so have images that have better contrast for the vessel networks, and also we're able to measure, for example, functional parameters like perfusion, like oxygenation, like blood concentration, we might be able to start to drive in and tease out a bit more about the types of disease that are present when we see them, and also how aggressive they are, going back to that early question that I posed you about early cancer detection. That challenge being that if you detect a lesion, you have to decide whether or not to act. And you want to act if that lesion is going to be aggressive. So there are already some advanced endoscopy techniques that do that. For example, narrowband imaging, where instead of the case in white light endoscopy, where we illuminate with a broad band of light between 400 and 700, in narrowband imaging, we're having a narrowband illumination at 450 and 550, which intersects with high absorption regions of blood. And what that effectively does is take an image like this, which is largely pink, and generates a colour contrast for the vascular mucosa. So what we thought is if we take this one step further and go beyond just the standard XY dimension with a limited colour spectrum and actually captured all of this spectral information, what we might be able to do is have that narrowband imaging data, which enhances the vascular structures in the tissue, but on top of that have functional information to really tell us something about the aggressiveness of the disease. So with that, I'm going to move on now to tell you a little bit about how we decided to tackle this particular problem. Anyone who's familiar with this idea of hyperspectral or multispectral imaging will know that it's quite a significant instrumentation challenge to capture not just spatial, but also spectral information. So to give a bit of background, the classic way that you might do this is you might focus a point of light onto the tissue and have that taken back and dispersed through a prism and detected with a linear array detector. So you have a 1D set of information collected at every single spatial point, and you simply scan that across the sample. Well, that's fine, but as you can imagine, that takes some time because you've got to dwell on each position for some period while you expose. And then once you finish, you've got to move along to another tissue. So it's not exactly a very practical approach in endoscopy where the patient is breathing, the endoscope is flexible, so it's moving all the time. We need to do something that's a bit faster. There are several other ways that we can do hyperspectral imaging. So for example, we can use a line scan where we take a line of illumination and we pass that through our entrance slit into a spectrometer, and we now disperse in 2 dimensions where one dimension is the spectrum and the other dimension is the spatial information. There we only have to scan in 1 dimension to capture all of the total hypercube of information. We can also, what we call sort of staring mode, so just look at the tissue in the full 2D piece of data. We can take a tunable filter that will look at one color of information at a time, and then we can tune through all the different colors in sequence that allow us to build up the spectral information now along the spectral dimension. Or we can try to do all of this at once in a sort of snapshot mode, where we try and capture all of the information. And in this case, it's quite a complicated snapshot mode where we're actually imaging along into different paths here, splitting up all the data into several different detectors. You can also do this by multispectral imaging directly by changing the color filter array that's placed on the detector that you measure. And I'll explain a bit more about that later on. There are also different configurations of endoscope that we have to consider. So in addition to these different sort of line scan and snapshot and wavelength scan imaging methods, we also have to think about how we're going to capture the optical data and bring it out of the patient. So we can do it in a chip-on-tip mode where you have LED illumination at the end of the endoscope. You have a camera that's placed at the tip and relays digital data out of the patient. And this larger channel here is an accessory channel into which you can introduce those biopsy forceps if you want to do any tissue sampling. Or we can have what's called a baby scope approach, where we take the existing endoscope and instead of using a digital relay of data, we put an imaging fiber bundle through an accessory channel, which allows us to relay light in of a particular wavelength we're interested in and also relay the image data out. And there are respective strengths and weaknesses of these two approaches. So for example, with the baby scope, we can much more easily move towards clinical application because these things are already approved for use in patients, but we have a very limited spatial resolution because the number of fibers that are in our imaging fiber bundle are typically on the order of 10,000. So we massively reduce our imaging spatial resolution compared to a chip-on-tip camera, where we can typically have at least up towards megapixels in terms of our capture, at least a few hundred by a few hundred pixels in our array. So first, I'm going to tell you about how we integrated some spectral capability into a baby scope approach, and then I'll come back to how we're thinking of doing that in the chip-on-chip mode. As I showed you, there's many different ways that you can go about doing spectral imaging, and we decided if we wanted to maintain both the spatial and the spectral resolution in endoscopy, what we needed was to go with a line scanning approach. So this is somewhat faster than the point scanning approach, and we have the advantage that when the endoscopist is actually using an endoscope, they tend to move the endoscope around across the lumen to actually survey the tissue. So we could actually take advantage of that motion in order to build up our full spectral data set. So here's just a layout of how we designed the endoscope. So this is the endoscope coming back here. The back of the imaging fiber bundle in the baby scope is then imaged through an objective lens onto both a wide field camera and onto a spectrograph with a 2D spectral camera. So now the slit is being applied across the center of the imaging fiber bundle, and we're dispersing one of our dimensions is spectral and the other is the spatial information. And the wide field camera is used to provide us with a reference through which we'll actually reconstruct our hyperspectral cube, and I'll explain about that on the next slide. So this is what's inside the spectral camera there. And here are the different ways that you can illuminate. You can either couple in your illumination directly into the baby scope, or you can apply an external illumination to the sample if you just want to try out different light sources, for example. We then reconstruct these data by using the wide field white light images to compensate for geometric distortions. So you can imagine as the endoscope's moving around, we'll get some rotations, we'll get some magnifications and some translations. And we calculate the geometric transformation matrices between each of the successive frames in the wide field camera, and that allows us to reconstruct a wide field panoramic image. And essentially we then take these geometric transformation matrices and apply them to the spectral camera data so that we can reconstruct our X, Y, and wavelength spatial 3D data set. And here's some examples on the left of the wide field panoramic data, and here's the wide field hyperspectral data that's been reconstructed. We can then use this to quantify hemoglobin oxygenation. This is a tissue mimicking phantom that we filled with blood at different oxygenation levels and took imaging data from. And when we take a look at the measured spectra and do spectral unmixing to resolve the oxygenation level, you can see that these relate nicely to the partial pressure of oxygen that we measured as a reference in these tubes. We also then wanted to know whether this would work in a practical setting. Of course, when you've got a USAF test target, you've got lots of really high contrast things that you're looking at. When you scan across those, it's really easy to calculate geometric transformation matrices. When you're in the lumen, as I showed you earlier, it's a relatively low contrast environment. Would you actually see enough in the white light images to be able to reconstruct? What we did was use a veterinary gastroscope and introduce our hyperspectral endoscope through the accessory channel. And we combined that with a pig esophagus model. So this is a pig stomach here and a pig esophagus that we can then actually introduce our gastroscope into. And as we move our endoscope around, you can see the white light image in the background there, the slit that's placed across the top of it, which is dispersing the spectral image onto our spectral camera on the right-hand side. And here's the co-registration that we're performing. as we move around. And here's the hyperspectral data cube. So indeed, actually having the lumen present and scanning across that, which is essentially what you're doing in endoscopy, gave us a really strong structure against which to reconstruct. So before I go on to talking about the clinical applications of this, let's just sidestep for a moment and think about the chip-on-tip cameras. I already mentioned that in a conventional color camera, you can build up color data using a color filter array, but you can also advance these beyond a standard color filter array that replicates our eyes to a more complex multispectral imaging grid. And this is particularly interesting if you want to look at different approaches. With creating a tailored multispectral filter array, actually it's quite hard to do this with standard fabrication processes. If you have a dielectric stack or some color dyes, that would be the standard way of producing the standard color filter array. So for every one that you would want to have here, every different color, you would have to go do another lithographic process to generate this. So you would generate your first iteration in green, then you'd move your mask, put your different dye down, move your mask again, pick the next dye down, and gradually fill up all of the gaps. The fact that this is a kind of a standard process is fine, but if you try and do anything non-standard, then and you want to do, say, a 3x3 array, now you're going to have to do this 9 times instead of 3 times, and it starts to become more complicated. What we did then was think about how we could do this lithographic process of exposing and depositing the color filters, but without having to do 9 different steps to achieve 9 different colors, or n different steps to achieve n different colors. And that approach that we took was essentially generating Fabry-Perot cavities, which is what you see on the left here, where we vary the thickness in order to vary the wavelength of light that's transmitted. But instead of doing this 1 by 1, we took a grayscale approach to this. So now each of these different cavities that's being exposed here in order to generate a cavity thickness for our Fabry-Perot cavity, we're using this metal insulator metal cavity where we have a metal at the bottom, we have an insulator in the middle, and a metal that's capping it off. That will give us our color resolution. But we can deposit this all in one lithographic step, all in one exposure, because instead of using the standard dose curve in the standard way by doing a binary exposure, either 0% here or 100% here, we use the response curve of the resist as a function of dose to tailor how thick this insulator deposition is, so how much resist is left in our cavity. And what that essentially means is that if we choose different exposure doses here and we calibrate this, this response curve, and when we apply them and we do the optical transmission microscopy, you can see a color resolved approach here. And we do the atomic force microscopy at the bottom, you can see that the cavities that are being generated by these different doses are different thicknesses. What this means is that we can carefully tailor the different color responses that we want according to this resist calibration response curve in a single exposure. And this is quite exciting because all of the existing approaches, as I mentioned, are quite cumbersome to fabricate and makes it quite hard to test out customized cameras. What you can see here is a plethora of different customized fabrications that we've done, ranging from standard square color filters with red, green and blue and IR response in this case, slightly more complicated multispectral approach in this case, and then different shapes as well as different sizes of color filter. And what we're doing now is looking at actually depositing these on image cameras so that we can do our own multispectral imaging directly on image sensors. So then in the second half of the talk, so the final part of the talk, to then go on to thinking about where do we go from here? What are the future perspectives on these particular approaches? What are the future perspectives in more generally for optics in early cancer detection? And how do we have to think about the landscape as things move forward? Well, the first thing that I thought was worth highlighting in terms of future considerations is that we have to think about how we take these devices that we develop in our labs, these sort of biomarkers that we're discovering, and actually apply them in patients. The step over from lab into even just first in human clinical trials can sometimes seem like a huge gulf. So the step forward into full application in healthcare is like climbing a mountain. It's really, there's so many different things that we have to consider along this pathway, not least of which is will this device actually stand up when it's applied in the patient setting? We also have to consider things like the health economics. Is it actually going to be of value to healthcare systems to adopt this new approach? What's the patient benefit going to be? Is the device going to be tolerated by patients? Can it be easily used? There are so many questions to ask. What we did a few years ago was actually start to think about this more strategically and say, well, what are the common characteristics for successful devices? And we took inspiration from this first review here, which generated a biomarker roadmap for cancer imaging in general, but didn't address optical imaging in this review. And we actually then thought about this quite carefully and wrote a second review article, which is a perspective really just for thinking about optics in cancer. And what we did was we went through all of the devices that had made it into clinical practice for optical imaging in esophageal cancer, which is the example I've already shown you, but then also for skin cancer, which is an area in which there's been very large number of devices actually make it through to clinic, but very few actually used routinely. And we said, well, what is it that gets a device to be successfully approved by a regulatory body for use in humans, and what is it that takes it that one step further to being adopted by clinicians for routine use? And there were a few different characteristics that we came out that I'd like you to think about and also keep in your mind as I sort of go through the rest of the talk, thinking about practical translation. So the first is, what's your contrast mechanism? Do you need to inject a dye or other contrast agent to generate background, or can you do this just with endogenous contrast? If you can do it with endogenous contrast, that generally seemed to be an advantage in the devices that we looked at. And that was largely because when you need to synthesize a contrast agent, this requires synthesis under good manufacturing practice. It requires a good evidence in terms of toxicology, and it requires you to think about how you administer and how you resolve your contrast agent. So do you apply it topically? For example, if you're doing endoscopic imaging, you might just spray it into the gastrointestinal tract. Do you need to inject it intravenously into the person and allow time for that contrast agent to circulate? So the added challenge with the contrast agents was simply that you needed to think about the practicalities of the procedure, how you would apply it, and also how you would produce contrast agent at appropriate doses according to appropriate production standards. So there was definitely a smoother path for devices that were based on intrinsic contrast in those that we looked at in this study. The second is thinking about instrumentation. So obviously any new device that you want to apply in patients is going to have to undergo a regulatory approval. And in the EU at the moment, that might be a CE mark. In the FDA, that might be a 510(k) or other approvals process. But in both cases, you're going to have to demonstrate that your device is safe and increasingly demonstrate it has clinical performance, its clinical performance value. And that's the same whether you generate an entirely new device or if you make a minor modification of an existing device. One thing that we found quite commonly in the examples that we looked at was that those devices that were compatible with the existing standard of care had a much higher chance of going on to be adopted by the healthcare system. And that could be for a number of reasons, but what we thought was that maybe actually it's helpful to have something that's compatible with the existing standard of care, because then you can directly compare your new device with the existing one. And if you're trying to show that it outperforms, that has an advantage, but even if it's complementary, it has an advantage that the two can be used at the same time. Another area to think about for instrumentation is also robustness. So are you measuring a very rare event that requires very highly sensitive and precise instrumentation? Or are you measuring something where you've got a very high signal to noise ratio and your instrumentation can perhaps be simpler and more robust? Something to consider in terms of the translational pipeline, where your device is going to end up in a clinic being bumped around in busy operating rooms where there are lots of people coming and going. Another area is the expertise of the operators. Depending on the type of optical imaging device, it might be operated by a layperson, by the public, and I'll talk a bit about that in a later slide, but increasingly there are optical diagnostics that are integrated into smartphones, so any one of us could be actually operating one of these devices. We could also be operated by professionals in primary care, so at your GP surgery, or by highly specialised operators in hospitals who have had years of training in how to operate imaging devices. What we want to be able to do is get similar results to existing methods at least, and that might be that we get similar results and that our approach is cheaper, and that might be a motivation for clinical translation. Or we want to outperform the existing device, in which case the operator has to be able to use it at least as easily as they can and generate as high level of a knowledge that will lead to a high level of confidence in the measurement of the biomarker. It's not just the person who's taking the data. In some cases, the person who takes and interprets the data are the same, which is often the case in endoscopy. But in other optical imaging applications, it might be that the operator and the person who interprets or reads the images is a different person. So there we have to go from the raw data that's acquired by the operator, process it and interpret it. So an example that's quite interesting to me is the one of narrowband imaging that I raised at the beginning, where we're illuminating the tissue with very narrow bands of light and detecting back the contrast arising from hemoglobin. Even though that was quite smooth in terms of its process through the regulatory pathway, once it was present in endoscopes and people were using it, in order to actually generate some standards against which it could be measured, there still need to be huge multi-center studies, and we needed to have a very large number of people develop classifiers which they could then use to interpret that narrowband imaging data. And that international consensus took a long time to establish, but it is now possible to use that in diagnostic procedures. So one benefit of narrowband imaging is that it directly maps onto the standard of care white light. So people can, although they don't visually see the two necessarily at the same time, they might flick between modes and see one then the other. The type of data they're being presented with is spatially very similar to what they're used to seeing. It just has this new contrast component added. Obviously, it sort of goes without saying, but is rarely actually tested in a lot of preclinical or discovery device areas, is repeatability and reproducibility. And this has to be both across the devices, but also across the operators and across the image readers or the interpreters. That whole pipeline has to be repeatable in order to be able to be applied for diagnostics. And we also have to think about whether the method we're using can be co-registered with other modalities. In a lot of the optical imaging biomarkers that we might be thinking about, for example, this spectral data, that's not something that you can straightforwardly map onto anatomy unless you have a classification procedure or computational procedure that you can apply and that you can give a color map to. So sometimes the anatomy just isn't present if you're doing, let's for example, a point spectroscopy measurement, and you need to add that together. So what we need to be able to do is map the optical data that we're acquiring with these advanced procedures onto other modalities. And this might be of relevance, for example, if someone comes to have an optical imaging view and then is sent for later for another surgery, it'd be great to be able to map the optical data onto that surgical interventional imaging as well. So the sort of the culmination of this evaluation and identifying sort of these key characteristics was the development of an optical imaging biomarker roadmap that we thought would help to facilitate translation. So I'll just sort of run you through the steps in this. The first one is the discovery domain. So this is where we identify the biomarker and realize its potential for use. The next is the validation domain, where we're thinking about validation of what we're measuring. And I've given some examples here. The validation might be in tissue mimicking phantoms, as we did with the blood phantom. It might be in ex vivo tissue or preclinical model systems, where you choose a system that best modifies or emulates what you're going to see clinically. Then we have these different levels of validation: technical, biological validation. Technical being more about the precision and the accuracy with which you can measure the biomarker, as well as any bias that might be introduced, and the potential availability with which you can measure it. Biological validation is then erring towards more safety, tolerability, and sort of clinical decision-making. Does the biomarker that you measure accurately reflect the underlying biology to allow people to then make decisions in clinics, either to test hypotheses at the clinical trial level or actually to manage patients at the level beyond that? In terms of the spectral endoscopy I've already introduced to you, I'd like to sort of come back to that a bit. And we got to the point with this over the last few years where we've really gone through a very extensive range of different tests in ex vivo tissues, model systems, etc. And we felt that really the only test that we were going to get a definitive answer from, whether this was ever going to be useful, was to actually go to first in human. And the reason that we wanted to take that step is to think about how it would impact the procedure in the long term. I already introduced to you why it's important to identify these precursor lesions in Barrett's, and I just wanted to give you an example of what having that improved contrast would do in the context of the healthcare system. So people are normally coming in for their white light endoscopy and their biopsies. We're then taking lots of these tissues off to diagnosis by histopathology. That's going to be very expensive and time consuming. If they're negative, then the person just gets told to come back on their 3 to 5 year interval. But if they're positive, then they have to come back for a second procedure to have therapy in order to remove those dysplastic lesions. If we could improve what we call our red flag imaging, so our wide field imaging that would alert the endoscopist to the presence of dysplasia, then we potentially run the advantage of having fewer biopsies, so less cost and less time associated with the biopsy, but also the potential to completely remove this procedure, this therapeutic procedure here, because we could actually do, if we had enough confidence and we could do the pathology swiftly enough, we would have enough, we would actually have the ability to do the procedure maybe even on the same day or during the same procedure. So we designed a first-in-human study to assess the spectral biomarkers of early disease. It's called MUSE for multispectral endoscopy. And the primary aim was to assess the feasibility of imaging with a spectral endoscope. And the secondary aim was to evaluate the spectral imaging patterns associated with non-dysplastic versus dysplastic Barrett's esophagus. We used in this, as I mentioned earlier in the talk, a clinically translatable baby scope architecture to accelerate first-in-human testing, but ultimately we'd like to think about how we could apply these in the existing chip-on-tip endoscopes, because that's really the direction that the field is going, with a lot more low-cost cameras being placed at the tip of the endoscope, as opposed to these imaging fibre bundles being applied. This disposable polyspoke catheter, into it we introduce an imaging fibre bundle. The glass at the tip of the endoscope covers the fibre bundle. We then have an illumination input, and we have a working channel where we can introduce further biopsy forceps. So this was our study design. We used the baby scope to relay spectral information to an external spectrometer. In this case, we kind of had all of the equipment on a cart that sat next to the patient. And this is our baby scope coming down inside the standard endoscope just here. We have a display from the spectral data and our own white light endoscope. And the light travels down through the illumination fibre and back up through the imaging fibre to a standard spectrometer. So in this case, we simplified the system, and rather than having a line scan, we actually just had a point spectrometer. And I'll explain why that is in the context of the study design. So the trial protocol that we set up was to try to be able to co-register our spectra with histopathology data. So we had the endoscopist do their standard of care procedure in patients with Barrett's esophagus. And if they saw any suspicious regions, they would choose one and mark it with cautery marking. And they would also mark a healthy region for us to use as a reference. We would then place our polyscope, so our baby scope catheter, right up in the center of this region and gather spectral data. We would acquire a large number of spectra per region. They would then go back in with their standard forceps, and they would extract a tissue biopsy from that region and send it off for histopathology so that we can relate the spectral data we collect to the gold standard histopathology report. So this is the reason why we use a point spectrometer, because at this point we didn't really need to image in this region. All we needed to do was to collect the point information that we could relate to the point biopsy. What I want to highlight here is just that even in this relatively small number of patients that we studied, discrepancies can arise between endoscopy and pathology. So it just sort of emphasizes the point that I was saying, that we do really need a better contrast for these lesions. So we had one trial where an area that was labeled as suspicious turned out to be just Barrett's esophagus, and another one where it was control, it was actually turned out to be dysplasia. So these are areas that we have to really consider, and this is exactly the kind of thing that we're trying to improve. And we took these tissue spectra and we used them to extract both vascular architecture and functional information. So we fit a biophysical model to our data with oxygenation ranging from completely deoxygenated blood, as shown in this top panel, all the way down to completely oxygenated blood in this bottom right panel. And we also looked at the vessel radius as a function from very narrow vessel radii in the very thin plots here to quite large vessel radii in the thicker plots here. And we took these functions and fit them to our data. And what we saw is that how blood vessel size and volume really changed during progression of disease. in this case, we didn't see much change in oxygen saturation itself, but what we did see was a marked reduction in vessel radius during non-dysplastic Barrett's, and then a further increase on towards neoplasia. And this kind of relates to what we would expect to see from a kind of a pathological setting, where we see an increase in microvessel density in the epithelial layer as the microvessels penetrate upwards during the progression of disease. And those deeper submucosal vessels in the squamous tissue, so the regular healthy tissue, tend to be larger vessels, which is what we saw here. And consistent with the visualization of those vessels, we also saw an increase in the relative blood volume fraction going from squamous to Barrett's through to neoplastic tissue. We then undertook a second pilot study with our full line scan endoscope, this time in the colon with some collaborators in the US, to further assess the potential of using this imaging-based approach. And again, here's the cart next to the standard endoscopy cart. We took our wide field images that I showed you earlier with the slit across. We can reconstruct a panorama from those. We can make an equivalent panorama for our hyperspectral data set, and we can extract example spectra from that. And what our preliminary findings show in this case is that there's also a clear distinction between the normal colon and the polyp tissue, as well as the regions that's left behind after resection. And you can discriminate these using standard sort of classification-based methods. So then the way of the future. We're gathering all of this spectral data, but what do we do with all this spectral data? We've got to be able to process it fast enough that we can give feedback to the endoscopist to tell them actually whether or not to resect some tissue, whether or not to biopsy. And the key thing here is to think about how quickly you can apply sort of these models that we've taken to do quantitative data analysis, or whether it should be done in a more data-driven way, for example, with artificial intelligence. So at the moment, our temporal resolution, as dictated by the data acquisition times of our instruments, can achieve real-time operation. That's not a problem. But our data processing times are typically far longer. We need to think about how we can best do our interpretation such that we can feed back this spectral data to the person who's operating and interpreting the acquisition. Machine learning methods are widely being explored for spectral approaches, and they've already been used extensively in other areas for hyperspectral imaging, like in remote sensing. But what could we do that would actually be useful from a biomedical standpoint? We need something that can do really fast online spectral classification. So we did actually look at our data set that was taken from the Barrett's esophagus patients in the upper GI tract with a convolutional neural network-based approach, training in for three-way discrimination between squamous, non-dysplastic Barrett's and neoplasia, as well as for two-way discrimination between different tissue types, because essentially what we're interested in seeing is neoplasia in the background of non-dysplastic Barrett's. In these cases, our accuracies were not too bad, but as we go towards identification of neoplasia, the sensitivity can be relatively low. The good thing is the specificity can be very high. And that's kind of, from an early cancer detection point of view, it's very important to have high specificity. You want to be able to give a confident no if you're going to send someone home and say you're cancer-free. If you're at all concerned about the patient, you always want to send them on for further investigation. So you'll want to biopsy the tissue and have some pathology done and that kind of thing. But I think as we gather more data from these trials and sort of boost our knowledge of how the spectra evolve between different disease types, I hope that we'll be able to apply these types of classification methods and manage to boost further both the sensitivity and specificity. We also took this one step further, particularly just using some very simplified approaches in terms of color classification, saying, well, we're not just necessarily going to classify the tissue type, so we don't necessarily have to build up a huge bank of data that says, allows us to classify between the different stages of disease. We could also use the approach that the endoscopist uses already, which is simply to look at the different colors of the tissue together with the morphology and make a decision about whether that tissue is suspicious. So we also took an approach where we said, well, let's train on color chart data, let's train on reference colors and see whether we're able to use those colors to classify different tissue types. And this is then how the data could look in real time. You could have your slit across your endoscope field of view, and you could get a real-time classification as you sweep your endoscope back and forth across the field of view. So that's something we're also looking into, and we're interested because in our early analysis of some ex vivo data, we actually were able to show that the colour classification did discriminate between different tissue types. This is something that we're digging into a bit further. How else might things move in the future? Well, obviously, I've already spoken that there's likely to be a role for data-driven machine learning type approaches in processing our data, such that we could present data to the clinician in real time and that they would be able to interpret that. But another area that's quite important is to think about where optical diagnostics will find itself in distributed healthcare. What do I mean by that? Well, I mean thinking about where healthcare will be done in the future. If you go from left to right here, we're just going to think about the current pipeline and how things might be applied in future. Our first column is people at home, second column is in primary care, third column is in specialist care. So our current Barrett's esophagus procedure is that our patient goes to the primary care centre with a GP, gets diagnosed with Barrett's and then gets referred for endoscopy. They'll undergo their diagnostic imaging, biopsies will be taken, targeted by imaging as well. They'll go for histology and then they'll go through to endoscopic therapy. What could happen in the future? Well, if we think about our primary care centre, we could already have diagnostic imaging happening there. There are many exciting new advances in miniaturizing endoscopic technology to applying capsules, which can be swallowed, and those capsules can survey. These capsules can be free, so pass through the entire GI tract, or they can be tethered, so just for application in the upper GI tract. And that allows us to map out the optical contrast throughout the GI tract into essentially a video that can then be interpreted offline or potentially even interpreted by a computer. Having done that procedure, we might then say, okay, this person is going to need some sort of treatment, and we send them to the specialist care centre, but now everything can happen in one visit. They undergo their advanced endoscopic imaging, they can undergo an optical biopsy rather than a pathological biopsy, so we could have a really high resolution endoscope here, which would allow an external expert to interpret those kind of pathology grade images, or a pathologist to interpret them. They don't have to be present at the site, they can be somewhere else in the world. We could even have the computer do an initial phase of processing, which allows the pathologist or the external expert to have a smaller data set to work with. So we reduce the number of visits that the person has to make to the specialist care centre, and we shift the process of care backwards towards the patient into localised settings. This goes even further in the example of skin imaging, which is something else we looked at in the review article I mentioned. In this case, at the moment, we might use the patient going to for their diagnostic imaging for visual inspection in the primary care setting, and then going on to the hospital. In future, they might use self-monitoring with their own mobile device, and that device can then look at changes in the colour and the shape and the features of the disease over time. And that lesion data that's provided there could be shared with their clinician via communications networks. It could also be further inspected with advanced imaging. And then the process for which you get to specialist care is relatively minimal. At the moment, when you get to specialist care, it can be quite complex in the sense that a skin lesion will be biopsied, sent for pathology. If it's positive, the person might be brought in for treatment. And in some surgeries, even the person stays on the bed while the pathology is happening, and there's an iterative process of pathology during that procedure. We could imagine, again, doing this all in one time with real-time feedback in a future setting. So more responsibility and more pressure placed on our home and our primary care centres. On the other hand, alleviating pressure from specialist care centres, which is how, at least in the UK, a lot of distribution of healthcare is going. We've seen actually quite a lot of this recently during the COVID pandemic, and I really point you to this review that was written by Darren Robler, who's published in Journal of Biomedical Optics just a couple of months ago, which was looking at the remote patient monitoring and the combination of home monitors, wearables and telehealth, particularly in the context of COVID. So with wearables, we're thinking about physiological monitors worn on your body, that are in contact with your skin typically, and that move with you in your day-to-day life. These are typically for preventative measures, they're superficial and they're non-invasive. So we're not going to replace an endoscope with one of these, but we might replace an inspection of our skin, or we might replace looking at global changes in our bodies as a surrogate for the emergence of poor health which could then be looked at in more detail for signs of, for example, cancer. These will monitor things like heart rate, pulse oximetry, often multiplex many different measurements, so they might need different spectroscopies together, optical spectroscopy with reflectance, but maybe also with fluorescence or Raman or other techniques in the future when we get to things being really small and miniaturized. There are also monitors that are implantable, so these are often therapeutic, organ-specific and invasive. For example, a blood glucose monitor or things that monitor metabolism or tissue hemodynamics. They can be used to look at tissue health after surgery, and particularly after transplant surgery for out of interest. So these home monitors, these home medical devices, there's been a massive increase in the use of these during COVID, and I actually saw some statistics that more than 26,000% increase in the northeast of the US in the use of telehealth. So actually, the idea of patients being in their homes and being able to be part of the healthcare system is no longer a future pipe dream. It's actually something, it's a reality that's happening day to day with us. To the extent that the FDA even relaxed enforcement of the use of some of these monitors that are typically medical devices used in a clinical setting, and allow them to be used in a home setting, for example, with oximeters and ECG. So I think this is a really interesting shift, and particularly from the optics perspective, because many of these existing wearables and smartphones are using light to capture information about our tissues. If we think about early cancer detection using optics, this is clearly something that's going to influence how the field is going in the future. If we parallel that with the emergence of new blood-based biomarkers and urine-based biomarkers, people that are looking at assays that you can test in things that are being excreted from the body, and combine these two ideas together, we can really start to think about a different future where we're monitoring well people and looking at how their changes in their well-being will influence, sort of change systemically, could influence their cancer risk, and potentially also give us an insight into the emergence of cancer. And this is something that my former mentor, Sam Gambier, who sadly passed away last year, was really a strong proponent of, thinking about the future as one of precision health rather than one of patients who are symptomatic being treated. Let's catch people before they get to that stage and try to understand how we can detect cancer at a much earlier level. So I think this is quite an interesting approach, and I'd encourage you to look at this paper and also the recent paper from Sam's team on the smart toilet that is able to carefully monitor our daily passages for signs of early cancer. This is an interesting way of the future using optics. So with that, I come to the end of my talk. I hope I've convinced you of the power of optical imaging in early cancer detection, given you an example of how we're developing a translational approach to spectral endoscopy, told you about some of our first-in-human testing, and then given some sort of broad perspectives on the use of optics in early cancer detection and where we might be going with the field in the future. So with that, I'd like to thank everyone in my team who's been involved in the research that we've done, and all of the collaborators that have helped us, particularly with the clinical trials work, Massi DiPietro and Rebecca Fitzgerald, who we couldn't have done it without, and Liana Sakitis and Sarah Diamond from OHSU for the colon work. And I'm happy to participate in the discussion that's coming up. Yes, from my side, I would say just
let Sarah finish, because she. I'll give you a very quick answer from my point of view, which is I think one of the fantastic benefits of lockdown is that I've probably attended more webinars and lectures and conferences in the last 6 months than I did in the previous 6 years, because I didn't have to travel and I could do it from home. And so doing these relatively short and focused seminars means that people can get a snapshot, can get a view of the whole field very, very quickly. I found it's been, it's annoying to be locked down, but it's been really helpful in widening my interest. I'll shut up now.
Funny, Dave, I've had a completely the opposite approach, because having a toddler at home means you never get a chance to see anything unless you get away from home.
Sorry, very male-centric.
Yeah, I mean, I think from my side that it's a similar response in the sense that I feel like, don't be afraid to just immerse yourself in another world. So if you're an early career researcher and you truly want to understand how to apply your physics or your engineering or whichever discipline you're in, and you want to understand how to apply it to biomedicine, don't be afraid to go to talks and webinars that are completely outside of your field, to take advantage of what's online from many different places now to attend summer schools that are in completely tangential but biomedical areas, to really immerse yourself in the point that Andy mentioned earlier, which is the language. Being bilingual between kind of medicine and physics doesn't come overnight. It takes years of practice and years sitting on the internet, searching for definitions of terminology and understanding what someone else is the point they're trying to get across. They often use the same words, but mean a completely different thing than we do. So without actually kind of going to these seminars and spending time to immerse yourself in it, reading books about kind of anatomy and physiology, and in my case, cancer biology, it's really hard for you to make that jump. But it's a really easy jump to make if you commit yourself to it and you're not afraid to take that chance and learn more about how medicine operates and where the place is for optics or physics in biomedicine generally.
Excellent. Anything to add, Andy?
Well, I think one of the key things is to, rather than just applying for that next postdoc, think about what you'd really like to do and just go looking for the fellowship or that thing before it gets advertised. Contact people. We can contact anybody in the world really easily now. So I think just contact people directly who you want to talk to, find out what's going on before the job gets advertised.